All right, good morning, everybody. Got my pointer going here. So my name's Martin Lawrence. I'm in the Department of Chemistry and Biochemistry. I've been at Montana State University since the late 1990s. I'm struck because and one of the reasons I like to come to this class is because you guys are mostly freshmen, science majors, and I've had a number of undergraduate researchers in my lab over the years, well over 100. I've had more than 25 graduate students, most of whom have gotten masters or PhDs, who've gone on to nice careers in biotech. But when I look back and I look at the best students that have come through my lab in that period, it's not those guys that got the PhDs. It's actually been the undergraduates that have gotten into the lab and started doing work early and found out it's for them and didn't worry about not knowing enough or being in over their head and just decided to go for it. And by the time they finished their undergraduate careers here at MSU, they had really accomplished as much as many master's level students, and even some would have been well on their way to a PhD thesis. And then they've gone on from here to graduate school, and because they got into a lab and didn't just spend 2 or 3 hours a week, but really put a lot of time and effort into it because they enjoyed it, they wound up excelling in graduate school. They wound up being way ahead of their peers when they went to these top-notch graduate graduate level programs. And then when they got their PhDs, they were really outstanding PhDs, and now they're faculty members across the world. So when I look at all the people that have come in through my lab in the last 25 years and say, which ones are the faculty members now, which ones are making the difference, every single one of them was an undergraduate. So it's really cool to see you here. Hopefully you'll see something maybe in what I'm talking about, more likely in what all the other folks are talking about. But whatever gets your attention and gets you excited, I encourage you to get out there and go learn about it. Try to get your hands messy. Get into the lab and do things. Roll up your sleeves and go to work. All right. So I'll come over to the other side of the soapbox here and talk about astronomical numbers for a little bit. Now, obviously, COVID is a huge issue right now, and I think for that reason, you've already seen a lot of virus talks. And that's appropriate and that's cool. This one happens to be a virus talk too, but not really in the COVID sense, okay? But I do think viruses are really cool, and viruses are super important in biotechnology as well. But let's just start with some numbers, astronomical numbers. I think something like 1 times 10 to the 25th is an astronomical number. It's astronomically large. Most of us have no clue what that number really means. Even after 1 trillion, which is 10 to the 12th, we have problems. And we're talking an order of magnitude greater than 1 trillion trillions, all right? But just as an example, 1 times 10 to the 25th is the number of stars in the universe. So that's certainly an astronomical number, okay? How many viruses do you think there are on Earth? And maybe you've already seen this in some of the other talks. And the answer is 1 times 10 to the 31st. 1 million times more viruses on Earth than there are stars in the universe, okay? So that's absolutely an astronomical number. It's just mind-boggling to me that we have that many viruses on Earth. Do they all infect humans? No, very few of them infect humans. Most of them infect other domains of life. So you are a eukaryote. You're over here in this tree of life. And the eukaryotes are this small branch right here. And then you have all these bacteria and lots and lots of archaea, and the number of organisms, microorganisms that are out there is still expanding dramatically. People now that they can just go out and sequence from the environment are finding new bugs that we didn't have any clue existed. Probably hundreds every single day, okay? So where are the viruses? These 10 to the 31st viruses infect all of these organisms. Wherever we have looked on planet Earth and found life, we have also found viruses. They're just everywhere. And in fact, if you go into seawater, for every single-celled organism that's there, there's at least 10 viruses floating free around in the water. So the next time you hop in the ocean and go for a swim, I want you to remember that you're swimming in a sea of virus. Okay? Most of it won't infect you. Most of it's infecting the lower order eukaryotes and bacteria and archaea that are in those oceans. And so then we can ask, and I borrowed this question from this slide from Blake Wiedenhef's lab, but we can ask, if there are so many viruses, how does anything survive? And I think that Blake's probably talked with you already about prokaryotic antiviral defense systems, and there are a lot of them. So prokaryotes, meaning things that don't have a nucleus, archaea and bacteria, have a lot of ways of fighting viruses. They can block absorption of the virus by changing the proteins or mutating the proteins that are on their surface. They can block uptake or injection of the DNA, and they have molecular mechanisms for doing that. They can be very altruistic, and they can commit suicide, essentially, and say, okay, I'm infected with a virus, but I'm going to check out, and if I'm dead, then that virus can't propagate and it can't infect my relatives. All right. So that's known as abortive infection. And then there are also restriction modification systems. And this is the first place where antiviral systems were important in biotechnology. So people like Dr. Sands and myself, and probably you as well, because they're still used, will use restriction enzymes to chop up DNA and then knit it back together. And all of these restriction enzymes came from antiviral restriction modification systems in bacteria. And so there's a whole company called New England Biolabs, or NEB, that built a huge business on restriction enzymes. And that's all out of antiviral systems, okay? And now, more recently, we've found systems called Argonaut or Slicer and Dicer, and this is now used in biotechnology and a technology known as RNAi, or RNA silencing. And it's very, very common in many, many laboratories around here. This has been around now for about 25 years. And then more recently, we've learned about CRISPR-Cas starting in the, around 2010, 2005. And since then, it's really gone ballistic since it's been adapted for gene editing purposes. And I'm sure all of you have heard about CRISPR-Cas and gene editing. But one of the points here is that all of these systems are derived from antiviral systems that have been found in prokaryotic organisms. And so all of these biotech tools wouldn't be out there if people weren't studying these strange little microorganisms. Okay? So I guess one of the rules here is don't insist necessarily on studying something that has obvious applications. If you just go out and explore what nature has to offer, you'll find incredible things that we haven't imagined. Okay? So I don't want to talk too much about CRISPR-associated adaptive immunity at all. I'm just going to skip over that, but I'll say that we've been working in CRISPR-Cas for well over a decade now, since before it was realized that CRISPR-Cas could be utilized for gene editing. The reason that we started working on it is because we have worked with Mark Young's lab very closely on viruses from Yellowstone National Park. And we got very interested in virus-host interactions, and we started noticing that there were certain proteins, Cas proteins, that kept coming up over and over again in various contexts as we were studying these viruses. So our main interest really started on the viruses and then turned to antiviral stuff in the archaea. And these are negative stain electron micrographs of a number of viruses out of the hot springs from Yellowstone National Park and around the world. These particular hot springs are usually above 80 degrees C and oftentimes very acidic. Mark says it's akin to boiling battery acid, but there are still organisms that live in there, and there are those archaeal organisms. And like I said earlier, when Mark goes and looks and says, okay, there's life in these hot springs, even though it seems to be extremely hot and very acidic, boiling battery acid, he finds out viruses as well. And these are some of the viruses that he and others around the world have found. And you can see that they have really interesting shapes to them, shapes that you don't normally see with viruses. A lot of them have these lemon-shaped capsids. These are containers made out of protein and or lipid that have the viral genetic information on the inside. Viral genetic information can be RNA or DNA, it can be double-stranded, it can be single-stranded, it can be positive strand or negative strand in the cases that it's single-stranded. I'm not going to go into that, but a virology class is certainly something that you guys could think about taking, I think, easily by your junior year if you wanted to, and learn all these things. But usually when you look at traditional human viruses, eukaryotic viruses, plant viruses, they tend to come in either a spherical or icosahedral shape or a long, thin thread, and sometimes it's rigid and it's more rod-shaped. And so what's unusual about the archaeal viruses is a lot of them have these lemon-shaped capsids, and some of those capsids have tails on the end, like this one here. This is called an Acidianus two-tailed virus. This guy has a single tail right here. And in fact, these viruses will actually grow tails. This virus here starts out with no tail, and then as it sits at 80 degrees, it will actually grow tails, and the size of the lemon-shaped capsid will decrease. Here are some rod-shaped viruses or filamentous viruses. And then here's a rather boring icosahedral virus here, but we've done a lot of work with it. So I'm going to give you some examples of our work from this today. But this is perhaps here my favorite one here. It's called Ampuloviridae is the family. This is a Acidianus bottle-shaped virus. Looks kind of, to me anyway, a little bit like a champagne bottle. And it looks like one of them is actually popped and it's coming out here. But if you look closely, there are some little filamentous structures down at the bottom of these bottle-shaped viruses. And we don't really know what they're doing, but in some senses, this looks a little bit like this part of the tail coming down here. So these are just unique morphological features of these archaeal viruses, and the main point here, the take-home point, is they're very different, okay? And when you look at the genomic content of these things, they're very different as well. But let's take a look at this really boring virus, the icosahedral virus, in more detail. This actually did come out of Yellowstone. It was isolated from a hot spring that was about pH 3 and could vary in temperature between 72 and 90 degrees C. It was the first icosahedral virus isolated from an archaeal host. And these are negative stain electron micrographs here. And then we can do a process called cryo-EM single particle analysis, and we can get a 3D picture of these viruses. And this is a cutaway here to show that it has a blue protein shell, and it has inside it a yellow lipid monolayer, and then inside that would be the genetic information. In this case, it's double-stranded DNA. And so this is a map of the genetic information. It's about 17,000 base pairs in size, and there are 37 genes that are on this double-stranded DNA. And when you take the gene sequence for any one of these genes and blast it against the database, it usually comes back with a hit to other archaeoviruses, but at the time that this was discovered, there were no other discovered archaeoviruses. But even now, today, when it comes back to hits to other archaeoviruses, there's no known function for most of these proteins, all right? And so the question, and this is where we got involved, if this virus encodes for 37 different proteins, what are those proteins doing? And that's where we got involved. Things that they might be doing is that somehow this virus has to get inside the cell, and the proteins on the surface are probably involved in getting this genetic information inside the cell. Once it's inside the cell, it needs to take over the cellular machinery so it can replicate itself, all right? These are very selfish. It doesn't really usually care too much if the host's well-being is satisfied. What it really wants to do is take care of itself. Sometimes it likes to insert itself into the host genome and just hang out there in a nice, cozy, warm place, because outside in the hot springs, it's extremely acidic. But if it thinks that host is dying, it'll pop out of that and it'll replicate itself super quickly and make 1,000 copies of itself and poke holes in the membrane of that cell and get out. And that cell is like Swiss cheese and it's dead, all right? So STIV needs to be able to take over the cellular machinery and drive its own destination. It needs to be able to express its own genes and make viral proteins. And then it has to assemble those viral proteins into a functional virus. And then it has to be able to get out of the cell. And so I think we can conclude that most of these genes here and their gene products, the proteins, are involved in these kinds of processes. And so the approach that we took was to clone every single one of those genes. So that's an exercise in molecular biology, and you learn some nice molecular biology doing that. And then we would take the clones and put them into E. coli and have E. coli express the viral protein for us. And so we had clones of all 37, and we'd have, one day we'd have E. coli making protein 1, another day it would be making protein 2. And then we would purify that viral protein away from all the E. coli proteins. So this is an SDS Page gel. These are standards of known protein sizes. And this is a viral protein here whose molecular weight is about 16,000 grams per mole. And once we have that protein pure, we want to try to study it and figure out what it does. And one way we can do that is to determine the three-dimensional structure of that protein. Because one of the real postulates of biochemistry is that the structure of a protein is what allows it to do a particular function, all right? And these are known as structure-function relationships. And a particular 3D structure can accomplish a particular function. And you can just think of this, you know, you look at a screwdriver. It's the form of that screwdriver that allows it you to turn that screw. And you can look at one screwdriver and another screwdriver, and you can say, oh, these look identical, except for at the tips, where one's a standard screwdriver and one's a Phillips head screwdriver. And when you see that, you can say, oh, this one has to interact with a different kind of screw. And these are the kinds of things that you can learn about when you look at protein structures as well. You can, from just looking at it, you can come up with good hypotheses about what these proteins might do and how they might do their job. You know, is it a hammer? Is it a screwdriver? Is it a saw? And all these, there are analogies to these kinds of functions in biochemistry as well. There are well-known functions for proteins, and they fit into functional classes. So we crystallize these proteins, and these are just some examples of protein crystals. These are much smaller than they look. These are probably a millimeter in size or smaller, but you can see that they have very nice facets to them. And so it's kind of cool that we can take a protein from a virus that's expressed in E. coli and get it to crystallize. And then once we have crystals, and this is if we get crystals of a protein, that's a huge celebration in our lab. It's just a wonderful accomplishment. And then we can take our beautiful protein crystals and we can put them in an X-ray beam. And when we put crystals in an X-ray beam, here's a cartoon of a crystal here, and it's being hit with a strong X-ray beam, much stronger than what you would find, say, in a dental office. And then we can put a piece of film or a specialized electronic detector here, and this crystal will scatter the X-rays and give us an X-ray diffraction pattern. And this is a diffraction pattern because there's a crystal grating there. It's not an X-ray of the protein. It's not like each of these spots corresponds to the position of an atom or the size of an atom. But I don't have time to really go into the physics of X-ray diffraction. But the bottom line is that we can take this diffraction pattern and a whole bunch of diffraction patterns, because we'll turn that crystal and we'll collect diffraction patterns from lots of different angles. And by getting those diffraction patterns from all different angles, we can make a 3D reconstruction of that molecule. And the reason that we use X-rays is because there's a law in physics that says that if an object is larger, if an object is smaller than the wavelength of the light won't interact with it. If you know visible light goes from about 700 nanometers to 400 nanometers, red to blue, that says that with a light microscope using blue light, you can't see anything significantly smaller than the blue light wavelength, 400 nanometers. It actually turns out to be half that, so it's 200 nanometers. And there are Nobel Prizes that were awarded even in the last decade for finding ways to use technological advances so that you can, with a light microscope, see something less than 200 nanometers. And that's a whole area known as super resolution microscopy. And they're down to about 50 nanometers now in super resolution microscopy. But if you want to see an atom, that's 1/10 of a nanometer. And so we're 500 times longer wavelength than we need to be able to see an individual atom. So if we want to actually see atoms, we need wavelengths that are really short. And X-rays have wavelengths on the order of angstroms, which lets us see atoms that are on the order of angstroms in size. And so that's why we're using X-ray light to see the structures of these proteins, right? And what do we see? When we actually use this technique to see the atoms, we don't see a ball and stick model, because the ball and stick model is just, that's our own fanciful way of explaining things. What we actually see is a cloud of electrons, because that's what an atom is. It has a very teeny tiny nucleus about the size, if you think about a football stadium, someone standing at the 50-yard line is the size of a nucleus, and the other 50,000 seats in that football stadium are the areas that are filled up by the electron cloud. Right? And so we see clouds of electrons, or electron density, but we're smart enough as biochemists to know that where there are clouds of electrons, there must be atoms. And so this is an example of electron density over here, contoured in a mesh. And if we zoom up on just a section of this, we can say, okay, here's a shape right here, and here's a chain coming through here, and this must be a tryptophan residue, and it must fit in like that because there's a hole in the ring structure here. So we can build a ball and stick model into that electron density and come up with a model for that entire protein. And this whole process is called X-ray crystallography, or even macromolecular X-ray crystallography, because they're large molecules. All right. And so how does this process work to determine the 3D structure of a protein? You clone and overexpress your gene. You express the protein that's made from the gene. You do biochemistry on it to analyze it, and then you crystallize it. You put it in an X-ray beam, collect an X-ray diffraction pattern, do a lot of computer processing to get a 3D electron density map, and then you build a model into that map, and then you're ready to study this protein and try to, in this case, figure out what it does, but in other cases, figure out how it does it, or if it's a protein say from SARS coronavirus, figure out how to make a small molecule that might interact with the structure of that spike protein to keep it from causing fusion, okay? And so I have lots of friends that are doing X-ray crystallography in pharmaceutical companies where they're involved in drug design, okay? So there are all kinds of good reasons to know the structure of a protein. And these are just a few of the structures that we've determined for some of these STIV genes. They don't have very fancy names. We call them A197 or B116 or F93. This is the one you saw earlier. And these just refer to the reading frames of the genes and the number of amino acids that are in them. And I could go on, I could talk way too long about any one of these, and so I'm just not going to do that. But from those structures, I'll say that we learned that some are transcription factors, some are coat proteins, some are glycosylases that put sugars on the proteins that give the virus more thermal stability, all kinds of things. But we also have now not just protein 3D atomic structures of the individual viral proteins, we also have 3D atomic structure for the entire virus, okay? And this came out again from X-ray crystallography and a second technique known as cryo-electron microscopy. And so this may not look like an atomic structure to you, but it is. It's just because we've zoomed so far out that you can't see the individual atoms. This is a 60 million Dalton atomic model, right? So it's huge. You have to zoom into it. It's almost overwhelming. If we look, though, at one of these, what we call turrets, and we can zoom up on those, and this is the structure of a turret here, which are found at the icosahedral fivefolds. There are two different proteins that make up the majority of that, this dark purple protein and this light pink protein, and they're called A223 and C381, and I could tell you more and more about those as well. But I want to skip forward now and show you what I think is kind of a new emerging area in biotechnology, and that is, okay, we can get structures of proteins that are isolated from a cell, but what if we actually want to know atomic structure of things that are in a cell? How can we do that, all right? And so we did a little study where we asked, okay, how does this virus attach to the cell? How does that happen at the atomic resolution? Can we come up with an atomic model for how that happens? And we took cells that from Yellowstone that can be grown in the lab, and we took some of the virus and we put it in with the cells, and then we let it sit for a small amount of time, and then we put it in a microcentrifuge and we spin down the cells, and they're much heavier and they spin to the bottom quickly, and we poured off the supernatant, which had the unbound virus. And then we looked and we said, okay, now is that cell infected or not? And we found that after you incubate the cells for 10 minutes, essentially all the infection that you're going to see is there. So if you put a virus in with the cell and you wait 10 minutes, that virus is already attached to the cell, because if you spin the cell down and pour off the supernatant, that virus is already infected. So then we went back and we took these cells, put in virus, waited 10 minutes, because we knew that was the optimal time, spun them down, put them onto a cryo-electron grid or an EM grid, and then we freeze them in vitreous ice, and we look at them with a new super-duper cryo-electron microscope, and this one was in Germany at the time. They're popping up all over the place now, even though they're very expensive. There's even one coming to MSU later this fall. But at the time, it was one of the few places in the world we can do that. And this is what we saw. So here's one of these icosahedral viruses out here. And here's another one out here. And this is a little bit of the cell, Sulfolobus sulfatericus. And this is its membrane right here. And then this layer out here is a proteinaceous layer known as an S layer. And maybe some of you can see that there are some little hairy-like things coming off. That was a new discovery. That's some kind of a glycan chain coming off. And the only place that we saw these viruses up along the cell was when we had these filaments coming out like this. And we could look at other parts of the surface of the cell, we wouldn't see any virus, but we soon learned that as long as we went and looked at where these filaments were, then if we zoomed in, we could always see the virus as well. And here you can see the virus looks like it might even be attached to one of these filaments. And maybe this one as well, because you can see there's a little bit of filament running along through here, all right? And this is the kind of microscope that we use to do this kind of work. So if Mark Young gave a talk earlier, he probably was virtual, but this is Mark, and this is one of my buddies that I made friends with while I was actually working in Germany for a year, and Mark came over for a visit. And where's the microscope? This thing here is the microscope, okay? And Mark's tall, he's like me, he's 6'3", I'm 6'4", all right? But that gives you an idea of the size of this microscope. So that's how we get this picture here, but it's not a 3D model. We also can't do what I showed you earlier, single particle analysis, because these aren't single particles. But we can use something called cryo-electron tomography, where we actually take a tilt series in the electron microscope. And this is very much like tomography, X-ray tomography, or nuclear magnetic resonance, where again, you take pictures from a variety of angles, and then in the computer, you can use that to reconstruct a 3D image. And so we can do that here as well. And the image comes out, the 3D image comes out quite low and grainy. But this is like a Z-stack. We're going down through it. And these are sections through the virus. And now here's the cell over here. And I'm just going to come back. We're going again from the top down, virus, virus, virus. I'm going to play that again. Here's the different filaments going through. Now we're going to get down to the bottom of the Z-stack. And now we're coming back up. So you're going to see the same thing. Here's a turret, 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 turret. You can see the black turrets on the virus. Here's the cell. And then what do? And the answer is that, okay, now we can see those rough, crude 3D shapes of the virus, but we know that this virus has icosahedral symmetry. Okay, it has 60-fold symmetry, and we can actually say, all right, this turret is the same as this one and that one and that one. And so let's take these equivalent parts and average them together and put them back out. And essentially now that means it's like we've seen 60 copies of the virus rather than one. And when we average them, we get a much clearer picture, because there's a rule in physics that says that the accuracy of your measurement is proportional to the square root of the number of times you observe something. In other words, if I measure something 4 times and take the average, that's going to be a much more accurate number than if I only measure it once. And if you statistically say, what's the standard deviation? The standard deviation will go down by the square root of the number of measurements. So if I measure something 4 times rather than once, my accuracy should be twice as accurate. Square root of 4 is 2. So if I have 60-fold averaging, square root of 60 is close to 8. So I should have 8 times higher signal to noise. All the grainy gray noise should go away. And so when we take that average and put it back into the micrograph and color it so that you can see it, this is what comes out when we average both the virus particles and the filaments. And now you can see a much clearer picture of what's going on. But it's what's really there. And after this, we can leave that 3D grainy tomogram behind and take this nice tomographic average. And remember that we have a molecular model for the whole virus, an atomic model. You can see already it looks like it's interacting with the turrets though, right? And now we can take this model and bring it in, push it right into this and fit it into that density, and it fits just like a hand in a glove. Okay? And now we can dissolve away the other two viruses that we didn't fit it into, and we can zoom up on this molecular model for our virus, and now we can dissolve away the tomographic average, and now we have an atomic model for what was in that gray, grainy cryo-electron tomogram. And we can say, okay, we know that it's this C381 protein that we have the crystal structure of that's interacting with this filament. That's its job, is to attach to the outside of the cell and hold on to that filament. And it uses what we call the second and third domains. And they're almost like a hand that grips onto something. And that's how this virus is sticking onto this, all right? And so this is just an example of the kinds of things that we can now do in what I call structural biology, where we want to understand what's going on in biology, but at the atomic level. And there are a number of biotech applications of these things. But I don't think we have time for that. So overall, our lab uses techniques like X-ray crystallography and cryo-electron microscopy to study these archaeoviruses. I didn't talk about it much, but we use it to study archaeal CRISPR-Cas. We're not interested so much in trying to do gene editing. We're trying to understand how does that prokaryotic adaptive immune system actually work in these bugs living in Yellowstone. Maybe additional biotech insights will come out of that. It's quite possible they could, but that's not our major goal for doing it. Our major goal is just curiosity about how that works. I also didn't talk about it at all, but we're super interested in iron transport and iron homeostasis in humans. There are more than 1 billion people, that's with a B, billion people in the world that are anemic. At the same time, there are many people in the world that have too much iron. In fact, the most common autosomal recessive disease in the world is hemochromatosis, and the carrier frequency in people of Northern European extraction is about 1 in 10. And so if you're a white male, the odds are about 1 in 100 then that you could be at risk for iron overload diseases. If you're female, it's a little less. And for the population as a whole, it's about 1 in 300 to 1 in 400. And we have a wonderful way of treating that disease straight out of the 1300s. We put leeches on people and suck out the blood. Well, we bleed them anyway. But there are all kinds of good reasons to understand iron transport and iron homeostasis in more detail. So you can see that this is really, today I showed you really hybrid of crystallography and single particle analysis and cryo-EM. And it's really these hybrid techniques that are really going to drive structural biology in the future to give us highly accurate molecular models of entire cells. So 20 years from now, I think you'll see atomic models for entire cells. And once you have atomic models for entire cells, you can begin to do computer dynamics and figure out how those molecules move and how they do their jobs. And I think everything that we want to do in terms of engineering new drugs or new techniques is going to really go to the molecular level. All right. And then the last comment I'll make is that most pharmaceutical companies are already using protein structures to guide their drug discovery efforts. And so if you're working for a pharmaceutical company, you're going to be expected to know the basics of protein structure. So this is coming full circle to what I've said at the beginning. Undergraduate scholars have made a lot of really important contributions in my lab to our study of archaeoviruses. They're good projects for anyone interested in the molecular life sciences. They're also a good experience for those of you that are thinking about maybe going into medicine because it's related to drug discovery. It's a good experience for those interested in Zika virus or Ebola or HIV or influenza or whatever we're interested in 3 years from now, because there will be a new virus. Okay? And the skills that you would learn include basic molecular biology, biochemistry, cell biology. So if you're interested, come see us. You can see the protein crystallization and X-ray diffraction laboratory. We're also installing a new $3.5 million cryo-electron microscope, which is scheduled to arrive in a semi-tractor trailer in late November or early December. And we'll be installing that and commissioning that over the next 6 months as well. So with that, I think I should stop and take any questions that you have. Yeah.
How long did it take you to go from like discovering the virus to 3D model?
The question, I think, was how long does it take to go from having the virus to having a 3D model of the virus? Yeah. And so I'm going to take you at your word. I'm going to say that, let's say we already have the virus and we have a lot of it and it's healthy and it's good shape and we know how to handle it and study it. Because that can be quite the process right there. But I think that it's going to depend upon the virus itself. So determining structures of some viruses is going to be easier than others. Some are going to be more cooperative than others. But the first structures of coronavirus were being done within 3 months of people having enough virus. Those structures were completed. All right. Now, that was done by people that were real experts in what they're doing. So if you come in as an undergraduate and you start working on it, it's going to take you a lot longer because you've got a big learning curve to get up, but it's something that's doable. It's something that's also changing very quickly because the technology is changing so quickly. But I think that realistically, from the time you have the virus until the time you have a structure, if you were, say, a graduate student that was putting 20 hours, 30 hours a week into a project, you could have that easily within a year. Assuming that the virus was really ready for structural studies. So it's the kind of stuff that an undergraduate could think about working on too. Yeah. Another question?
Well, when I hear a basic talk, I always suggest that students say, so what? And you answered the so what question in many ways by saying it applies to coronavirus, it applies to all kinds of things, and drug companies are now looking at this as a way to find a silver bullet, if you wish, to stop a disease. So the so what question is important to ask in biotech, and you answered it. Any other question?
Yeah. We physically crystallize it. You might stop and think about how you would crystallize salt, for example. And one way that you would crystallize salt would be to just, start by dissolving it and making a salt solution to begin with. Get your Morton salt can out and dump a whole bunch of it into some water and heat it up. And whatever doesn't go into solution, just pour off the stuff and leave that at the bottom. And now you've got a saturated salt solution, or even maybe it's not completely saturated even. And then if you just take that pot and let it sit on the stove for 2 or 3 weeks, the water will slowly evaporate off, but the salt won't. And at some point that will become super saturated and the salt has to come out of solution. And if it comes out of solution, it will, and it does it slowly so that it can come out and not give you a precipitate. It will give you a nice ordered ionic crystal, because that's a lower energy conformation when you can have alternating positive and negative sodium ions put in a nice lattice. The Coulombic interactions are so strong that energetically that crystal form is much more stable than a precipitate. And the same thing is true of proteins as well. If we can find a way of drawing off the water in a controlled way, and we have some very clever but low-tech ways of doing this, then at some point the protein becomes supersaturated and it's going to have to start coming out of solution. Usually when we do that, it comes out as a precipitate, but if we find just the right set of conditions, pH, we need some counter ions, some positive and negative ions, because proteins have charges on their surface. If they're going to touch each other in a crystal form, you have to maybe, if there are two negative charges coming together that would repulse each other, if you have a calcium ion in between, then all of a sudden that becomes favorable. So we usually need some counter ions, and then we'll get these pretty crystals. And they're small, like I said.
Let's say you're an undergraduate and you walk into his lab. What you're going to find is you're going to learn not only from you, but all the kinds of people in your lab. Could you go into that for a minute?
My lab currently has 4 graduate students and 5 undergraduate researchers. And some of the undergraduate researchers are very advanced. In fact, one has a publication coming out in Journal of Biological Chemistry in 2 or 3 weeks, and she's the first author on that. Some of the graduate students are quite advanced. Some of the graduate students are just beginning, and some of the undergraduates are just beginning. And so we have people with really advanced skill sets, and we have people that are just learning. And usually I pair them up at the beginning so that you're working with either an advanced undergrad or a graduate student. Oftentimes they don't know that much more about it than what you're doing, and then I check in once a week or so and find out how things are going.
We have another class at 10 o'clock. You
Yes.
better run pretty soon.
All right.
Thank you very much.
All right. Good luck, everybody. And by the way, the human race will survive COVID-19 just fine. Global climate change is a much bigger issue.